Thank you for joining us for our monthly lunchtime webinar. We are fortunate today to hear from Dr. Pamina Gorbach, professor in the Department of Epidemiology. In this webinar, she will talk about the known roles of substance use in our local HIV epidemic in Los Angeles and point to the areas where mechanisms of enhanced transmission are still not fully understood. Following the lecture, we will have time for some questions. If you have a question, simply type it in the field at the bottom of the chat window in the bottom left-hand corner of your screen. Let's get started, Dr. Gorbach.
Okay, hello, can you hear me? I guess, yes.
Yes.
All right. I will then just go ahead and talk about this, and it's a great pleasure for me to have an opportunity to talk about substance use, HIV, and the research program that we are in the midst of just starting here at UCLA. So thanks for joining. I'm happy to take questions as I go. So if you'd like to, please go ahead and type in a question in the chat, and I'll handle it as I go along. Okay, so to advance this way, right. So today I'm going to talk about why it's important to study substance use in HIV among MSM, and MSM is the phrase I'll use to refer to men who have sex with men, which is sort of a generic term. It includes men that are gay identified and men that are not. I'm also going to talk about what I learned about substance use over the course of the last sort of 15 years I've been doing work in this area. And then I'm going to talk about how we're tackling substance use and HIV in future research here at UCLA through this program. So first of all, drug use in America or in the United States right now. There's a lot of people who use drugs. That's sort of a known fact. In fact, over 9% of adults were current users in the past month, according to the National Survey on Drug Use and Health. But if you look at this drawing, at this graph here, you can see that most of the drug use is actually this blue line, which is marijuana. So we've got a lot of people who use marijuana, and I think that as we move forward in time and more states are legalizing marijuana for both medicinal and recreational use, you know, this may disappear from this sort of graphic as it may not be considered illicit always. So we always had a lot of marijuana users. But then as you go down, you can see the numbers get a lot smaller, but this represents the general population. And so when you translate these numbers into actual numbers of people in the United States, the numbers become pretty large, which means that lots of people at some point use drugs in the United States. What becomes the issue is when they become dependent on drugs and become drug abusers. And those numbers go up even further and are even sort of more of a concern for us. If you look at this, this sort of shows in the past year how many people we have that are considered drug abusers in the United States. So the bottom line is we have a lot of people in the country who use drugs. Now, what about here in Los Angeles? Well, in Los Angeles, we have a lot of methamphetamine users. And it's interesting in the United States, when you look at the distribution of drug use, you see very different patterns geographically. You know, in the northeast of the United States and in the northwest, you see a lot more heroin use, for example, and injection of heroin than you do here in Los Angeles. For some reason, and some people have joked that maybe it's the sunshine, people don't seem to gravitate towards heroin use as much, and we've never had sort of a large population of heroin users. There are heroin users in the United States, and in Los Angeles, but what we seem to have more of are methamphetamine users, and that has been going on here in Los Angeles as a pattern for more than 10 years, probably close to 20 years. If you look at these data, which are from 2010, 2011, you can see that methamphetamine is accounting for a lot of the public treatment admissions and is the primary drug of abuse, in fact, for public treatment of admissions for about a third of those we see here. So lots of drug users in the country, lots of methamphetamine users here in Los Angeles. And so Why do we worry about this? Because this chart sort of summarizes what happens if you become a methamphetamine user. And one thing that this doesn't show is that methamphetamines are highly addictive, and people become rapidly addicted to them. And so all these effects then become much more extreme among people who are addicted to methamphetamine and become heavy users. And that trajectory is rather rapid for this particular drug of abuse. So what happens is you see these physical effects and psychological effects that are found in both men and in women. Methamphetamine has also been shown to be highly associated with certain comorbidities, in particular depression and ADHD and psychosis. So using methamphetamine is very interactive with these conditions. It's not clear if it causes them or people who have them tend to gravitate towards methamphetamine use. So what happens with methamphetamine use and where does HIV fit into this? Methamphetamine use, and we listed some of these here, but I'm going to talk a little bit more about the physical effects in a moment. But methamphetamine use results in or is associated with more stress, demoralization, CNS inflammation, subcortical injury, cognitive impairment. All these things become linked to depression and HIV. And the more you use meth, you also have impulsivity, senses of hopelessness, carelessness, demoralization, and cognitive impairment. And so why MSM and methamphetamine use? Because as I mentioned, there are women that use methamphetamine too. Well, we have a significant epidemic of MSM throughout the world. And in fact, this graph here sort of shows that throughout any region of the world where there is data on rates of HIV among MSM, you see that there are very high prevalences compared to the rest of the population of all adults among MSM. So the epidemic among MSM globally continues to sort of rage on. And here in Los Angeles, again, it also is the same pattern as we see in other countries. Most of our epidemic is among MSM. 85%, in fact, of diagnoses in Los Angeles County over this period of time where we have the data available from 96 to 2011, most of the HIV that we have locally here is among MSM. And it's not just among any MSM. In fact, what we see here in Los Angeles and we have seen throughout the country is that the epidemic among MSM is predominantly among minority MSM. And this shows the prevalence, which are the number of people currently infected with HIV in the population, and these are the newly diagnosed cases, so sort of suggests the incidence of HIV. And as you see in both of these, the highest bar is among African American or Black MSM. There was a study done that was part of a clinical trial that was in 6 sites throughout the United States through the HIV Prevention Trials Network, and it had a site headed by Dr. Steve Shaptaw here in Los Angeles at our UCLA research clinic at Vine Street, which was called the Brothers Project. And in that particular study, which was just completed about 2 years ago, the incidence of HIV among Black MSM was almost 7%, which is extremely high. So we have a big epidemic of HIV among particularly minority men who have sex with men here in Los Angeles. And we have also a lot of drug use, particularly methamphetamine drug use here in Los Angeles. So because of that, we started to sort of see how do these things fit together. And so I'm going to sort of show you some of the evidence that there is about methamphetamine use and HIV, and then I'll talk to you a little bit about my own work on the subject. For a while now, probably more than 10 years, we've known that there is a very strong association between use of methamphetamine, reported use of methamphetamine, and acquisition of HIV. And this has been shown through a number of studies and in different settings. So both in cohort studies like the MAX, which is the multi-center AIDS cohort that I'll talk a little bit more about later, we showed that methamphetamine is associated with HIV acquisition. In STI settings, this was shown. And then in a cohort that I worked on myself of newly infected men who have sex with men that started in San Diego, and then I continued in Los Angeles. This is my doctoral student a number of years ago. We showed that men with newly infected with HIV were more likely to be meth users. So clearly we saw meth use correlates with sort of a 2 to 4 fold increase in risk for HIV transmission. In my own work, in that cohort, I looked at how meth was sort of driving ongoing risk, not just that acquisition of HIV to begin with. And so what I did was I followed men who had been newly identified with HIV over a year. And what we saw is this is months since their baseline, and this is their pattern of transmission risk. So their likelihood to sort of transmit HIV to others. And what you can see is that their risk remained pretty high over the years. And in fact, when you look at their HIV status unknown partner or negative partner, while initially we saw a downward trend in reports of unprotected sex with such partners, it went back up after a year. So when I looked at why did it go back up and who did it go back up with, we could see that it went back up with, mostly it was being driven by these methamphetamine users. And so those who used methamphetamine at baseline and those who were still using it a year later were much more likely to report having sex, unprotected anal intercourse, so that's high risk of transmission, with their last or previous partner after an entire year after they'd seroconverted. So methamphetamine use is associated not just with acquisition of HIV, but is associated with ongoing transmission of HIV. So these findings that I've just reported are showing associations between behaviors that people who use methamphetamine use, or men who use methamphetamine use, and HIV. Now these are behaviors, okay? So we know that they're practicing these behaviors. But a number of years ago, along with some colleagues here at UCLA, we became interested in what is behind those behaviors. What are sort of the biological processes that are driving those behaviors? And back in 2003, in fact, there was this very interesting paper that came out that shows that actually methamphetamine use increases HIV replication in humans. And what this shows is they did a study both of people who used to use methamphetamine and were currently used methamphetamine. So the tox positive refers to people who tested positive in a toxicology. That means they had acute use of methamphetamine and also reported being on highly active antiretroviral therapy for HIV. Now what this shows is that people who were tox negative, those were former users of methamphetamine, they weren't current. You know, they are, and they're using HAART, you can see they're well controlled, their virus is well controlled, and there's lower replication of HIV. However, those who were current meth users, even in the presence of HAART, show a much higher replication of HIV. And so this is a finding that really became of interest to us because we said, you know, for most things, once people are on therapy for HIV, you see sort of a washout of effects of other things. However, methamphetamine still seems to be having a sort of a background effect on HIV replication in the system beyond, even in the presence of therapy. So I looked at this again in other ways. I'm sorry that the graphic for this didn't come out very clearly, but this is from a paper that was published in JADES a number of years ago, and online for some reason it has a poor quality. But basically I was interested in what kinds of HIV people were acquiring when they reported methamphetamine use. And what we saw was that people who acquired a resistant virus that was a resistant form of HIV were more likely to have reported recent methamphetamine use and polysubstance use. And so this was very clear when we looked at this in the first cohort of men with new HIV from San Diego, that methamphetamine use at that time seemed to be really driving not just the HIV epidemic in general, but also the spread of resistant virus in this population. So following that, with some colleagues here at UCLA, Dr. Steve Shapta and Peter Anton, we decided to try to look at methamphetamine use and how it affects the gut immune activation in men who have HIV but are not yet on therapy. So this was one heck of a study to try to do because that meant that we had to find HIV-positive men not on therapy who were using methamphetamine but nothing else because we wanted to really isolate the effect of methamphetamine use. So we ended up with a very small sample size, although we screened over 80 men trying to enroll active methamphetamine users who don't report use of anything else is really a tough thing to do. But we did manage to do them, and by the way, they all received a biopsy of their gut immune tissue, so that of their gut tissue, so that meant they had to get a rectal biopsy. So we had to bring them over to UCLA in the morning. Methamphetamine users, yes, get up in the morning, come over to UCLA, lie down, and get a rectal biopsy. So that was another reason why it was particularly challenging to get people to come and participate in this study. But when we did, we found something very compelling, which was that we can see these are immunological markers right here that came from the rectal fluids. These are immunoglobulins, and these are some cytokines. And what you can see is that among the meth users, we saw greatly elevated rates. Okay, so this is no therapy, but we did see that in this particular rectal tissue, HIV seems to be affected right here in the presence of methamphetamine use. The rectal immunology is greatly affected. So we thought, wow, this is a very compelling finding, but this was a tough study to do. We had small numbers. So we seem to be sort of scratching at the surface of a bigger problem of how methamphetamine use affects the system of people who have HIV, not just their behaviors. So because of that, we brought together the team of people that have been working on this issue here at UCLA, and we're able to apply to the special program that the National Institute of Drug Abuse just started to support cohorts long-term, enroll them and support them to look at the effect of drug use on HIV. So we were really pleased to be the first new cohort funded in this group. We have a very impressive group of colleagues in it. There's only 5 of us in the consortium right now that NIDA is supporting. 2 of them were 3 of them actually were long-term cohorts that have, and I'll show some of the findings later, made some really significant and substantive contributions to our understanding of HIV. And these include the ALIVE cohort of injecting drug users at Hopkins. It's over 20 years old. The Moore Clinic, also at Johns Hopkins, it's also over 20 years old. And the CITUS cohort up in Vancouver that's also close to 20 years old. So we're sort of the new folks on the block. Chicago also has a site focus, but we are sort of unique in that we are interested in studying sort of non-injecting drug use, since 3 of the other sites are doing injecting drug users, and also really doing an integrated study that goes beyond sort of descriptive epidemiology, goes beyond understanding just behaviors, then looks at sort of the interaction of the biology and behaviors for substance use on both people with HIV and without HIV. So let me tell you now a little bit more about this study. It is a new cohort in Los Angeles that we started enrolling on over a year ago. We are interested in looking at how substance use is linked to HIV transmission in the United States. As I mentioned, people who inject drugs are well studied, particularly those who inject heroin. But there were no cohorts of non-injecting substance users but when we started this. We also, as I showed you, minority MSM are disproportionately affected by HIV both in Los Angeles and in the United States in general, with higher incidence and prevalence, but few behavioral differences have really been noted between minority MSM and other MSM, which suggests that there is something going on beyond and behind the behaviors. So that is why in this particular cohort we felt the need to examine how biological factors are driving transmission in coordination with network and behavioral factors. We also sort of tip our hats to the MACs, and all of us have been part of the MACs that work on the M study so far, or most of us. MACs is the multi-center AIDS cohort, which has been going on here at UCLA for over 25 years, but is winding down in the next few years and will be closing by 2019. And so we will be following in their footsteps, and we sort of see ourselves as the son of MACs. And finally, we were very proud to call this the masculine study because that stands for an acronym. But we found in our early work with the community that that wasn't a very that title sort of alienated some members of our community who don't consider themselves masculine. So we have changed the name to M study. So officially to NIDA, we might be known as masculine because it stands for Men and Substance Use Cohort at UCLA, linking infections, noting effects. But the name masculine isn't something that resonates with our community, so instead we use M study. So let me tell you a little bit more about it. So our objectives are to establish a research platform to support the direct study of interactions between non-injecting substances of abuse infectious disease acquisition and HIV progression. So I want to point to the fact of this term here, research platform. That means this is not just a single study. What we're establishing is the infrastructure for some of our own work, but also to encourage the work of many other types of scientists, both here at UCLA and across the network of people studying HIV, to look at some of these factors in our sample. So we will do that by building a cohort of non-injecting substance-using minority, Black or Latino, MSM, who will be HIV positive with a viral load, and HIV negative at high risk for HIV infection. So those are the two main groups, and I'll show you a schematic of this in a minute. So we are building this platform for biological and behavioral questions of interest for substance-using minority MSM to provide opportunities to evaluate structural and functional correlates of biological and immunological deficits. And finally, it will allow the separation of biological versus behavioral and contextual influences on the observed outcomes among active substance use using MSM, both HIV positive and HIV negative. So we have main aims for the platform in the first 5 years that we're funded. The main aims are first to determine how and whether stimulant and other substance use impacts ongoing HIV/STI transmission among poorly controlled HIV positive and HIV high risk minority MSM based on their mixing patterns and exposure networks, which we will describe behaviorally and phylogenetically, the practice of transmission behaviors, adherence to HIV therapy and biomedical prevention, and mathematical modeling. So our overarching hypothesis is that non-injecting substance use is a key driver of the epidemics among men who have sex with men in Los Angeles. 2, we're going to assess how the gut mucosa may be affected by stimulant and other substance use affecting HIV susceptibility and pathogenesis. So we will characterize the altered vulnerability to HIV infection of the rectal mucosa in substance use and determine whether substance use more directly enhances HIV replication, shedding, and mucosal inflammation in HIV positive MSM. And we will be establishing and have started to establish a unique biorepository to do this. So here's what our plan is for the first 5 years that we are funded. Our goal is to recruit and enroll over 1,000 men. We expect that at the end of 5 years, we will end up with retention of about 620 of these men. So that means that we assume a baseline measure taken on 1,000 men, but given the high rates of incarceration and difficulty with follow-up among substance users, we expect to have retained 620. So half will be HIV negative, half will be HIV positive, half within each of these groups will be substance using, and then we have a comparison group of non-substance users in each group. The retention we expect among the negative men to be about 80%, and among these HIV positive men about 50%. And this is a conservative estimate based on our previous experience. As I mentioned, I have run a couple of cohorts among newly HIV infected men who have sex with men, and I have learned that they're very difficult to retain in research studies. So this gives us sort of some room to move. We have expected an incidence of about 6%, which would give us about 25 seroconverters. And then we have planned a sub-study that will involve biopsies of rectal tissue that would be done among a subsample. So what are our recruitment strategies? That's a lot of guys we expect to enroll, right? We've been working on this. This has been our first year of sort of being fully up and running. And in this time, we've been working a lot on getting our procedures in place to make sure that anyone we enroll has their samples collected properly and that the data is secure. So we've had a lot of work to do on building our infrastructure. And we are also doing a lot of work with the community to try to figure out the appropriate messages and the right way to reach people. These are examples. They're not final. Copies of some of the recruitment strategies that recruitment materials that we've been developing with our community sites and our community groups. They are reflective of sort of a party lifestyle, as you can see. They're trying to have imagery that's very typical of Los Angeles. We have this new logo, Turn Up, for your community. And this was developed after extensive marketing research. And we've started doing some online recruitment already, but we'll be looking into more innovative and interesting forms of online recruitment beyond sort of Craigslist as we move forward. So what do we do when we actually do successfully recruit and enroll somebody? This is an example of the assessments, a list of the assessments that we collect at baseline, and a subset of these are also collected at every follow-up visit. All of these are collected on an annual basis. We have though visits that are every 6 months. So at the 6-month visit, we tend to have a collection of a little bit less than the complete set here that you see. So we always have a behavioral questionnaire. It is web-based, and it's done through computer-assisted survey interviewing. And then these are all the measures that we collect. And I'm going to not read them all individually because they're long. And guess what? I have more. But I wanted to point out in particular that we also have biomarkers for substance use. We do a tox screen on urine, but we are also collecting hair, and nails, and I didn't have a good picture of nails, but those are ways, hair and nails, of assessing longer-term substance use. The urine tests only assess acute use of substances, like in the past 3 days. The hair and the nails can detect substance use patterns that go back 6 to 9 months. So we're collecting both kinds of biomarkers in addition to self-reports. In our behavioral questionnaire, we cover a lot of different topics and categories. And so this is an example of some of them, but I wanted to point out that we have a couple of them sort of well studied scales. And we are measuring things like depression and substance use on the behavioral questionnaire. And then in terms of what we collect for specimens in our biorepository, we do collect blood. And from the blood, we are extracting PBMCs, which are being stored in our repository, and serum, and hair I mentioned, and nails. So we also will have saliva, rectal fluid, 2 sponges, and a rectal swab. And then these are things, this is part of the blood. So quite a lot of specimens from each individual that go into our biorepository, which is going to be frozen at -70. We also have these mucosal immune measures that we will be running on a regular basis on these specimens, and these are listed here, and the specimens that they will come from are listed here. So who is our team and how are we actually doing this? So this is a little small, but I want to start actually at the bottom and explain that this study is actually being done in the community at 2 community sites. We have a UCLA community research clinic that's directed by Dr. Stephen Shoptaw, and that actually was begun over 10 years ago through another NIDA U01 that Dr. Shoptaw and I were co-PIs on called the SAPCAP study. And so we started realizing when we wanted to study substance users and HIV in the community at that point that we needed to have a community site that was more friendly to our participants. So this clinic was started back then in SAPCAP, and Dr. Shoptaw has continued to use it for many other studies since. And it's a wonderful resource and an excellent sort of community site. And we will be enrolling to HIV and are enrolling HIV negative men at this site. It's called the Vine Street Clinic, so it's on the corner of Hollywood and Vine. And it's all UCLA staff, and it's on the UCLA backbone in that clinic. The other site is the Los Angeles Gay, Lesbian, Bisexual, and Transgender Center that I have been doing ongoing research with for the past 15 years and has been a tremendous community partner for all the work I've done personally and many others here at UCLA and in the community have done. Anyone who knows the center knows that they are a huge community resource for men who have sex with men, and actually the gay and transgender community in general, as a site, an STD clinic, publicly funded STD clinic, as an HIV clinic, and as also sort of a general social resource clinic for the community. So this is a tremendous community site. It's very busy and very active. There's always a lot going on there, which sometimes makes it a challenge to do complicated and difficult research at, but they are a wonderful community partner and resource, and we're really pleased to have them as the site for enrolling all our HIV-positive men at this time. We have formed a number of committees, and I wanted to just mention some of the UCLA faculty that are part of this research platform as investigators. As I mentioned, I have a co-PI who's Stephen Shoptaw from Family Medicine. Leading up our our data core is Dr. Ronald Brookmeyer from Biostatistics. Our biorepository director is Peter Anton from Medicine. Our study director is Amy Ragsdale here in Epidemiology. We have Dr. Robert Dennis over in Medicine as well, who's the head of our data management group. We have other investigators like Tim Hall, who is in Family Medicine, Marjan Javanbach from Epidemiology, Naina Harawa from a number of departments on campus, including Epidemiology, Mark Malik, who's in Epi, and also now we've recently Jesse Clark from Infectious Disease has joined our group as an investigator, and a wonderful team of other folks all throughout campus who are essential to running, and at the sites, this very ambitious study. So I'm sorry I didn't list everyone's name, but they're all listed on this page. We do, as I mentioned, have a biorepository. We do anticipate about 240 subjects per year being enrolled, and that is just the enrollment, and there will also be those doing their follow-up visits, so this repository will grow over time. And so we are establishing and looking into both resources on campus for long-term storage as well as some biobanks off campus, but all the specimens will be stored for future use. And then the bioinformatics. When you start a study like this and collect all this kinds of data, one of the challenges is trying to integrate the data, because sort of one of the key themes of this platform is the integration of science across different disciplines. So in order to do that, I mentioned Dr. Dennis from CTRL, which is Computing Technology Resources Laboratory over in Medicine. He has been working with us to design this very complex system which serves as a web-based online data entry and data integration system. So as you can see in this, there's all sorts of systems. The basis of it is a system we call P-Tracker, where the data is entered at the sites. The data comes in from the computer-assisted interview. The data comes in from all different places, and then eventually all the data will go to a data warehouse here, including the biorepository specimens. So we've been really excited that even though we are just finishing the end of our year 2 at the end of the spring, we've already had a lot of interest in the research community to work with us. And so we're thrilled, because that's our job, is to be a platform and not just do the science ourselves, but to work and support other people to do their science on this. And so one of the most exciting pieces that has come out of this is a supplement on studying the brain through conducting functional MRIs of the brain of substance users. So we will be, we have submitted a supplement, a large supplement, to our main study that would be headed by Dr. London over at NPI here on campus. And it says resubmitting in December, but actually it was resubmitted in December and is being reviewed this month to integrate sort of studies of the brain into the other research we're doing on this project. So the effects of methamphetamine on the brain is another whole lecture, and Stephen Shoptaw on campus here gives a great lecture on that pretty regularly, so make sure to keep your eye out for that if you're interested in the effect of methamphetamine on the brain. But we're excited to be able to hopefully integrate that into this into our research program already. We have another of other concepts that have been submitted for research that are in process, including an exciting one on criminal justice involvement for MSM that looks like it is funded, that Dr. Harawa is working on, and some other things that are in various stages of development. So I wanted to just sort of end with the types of contributions that cohorts like this can make to the field and what we hope to be able to do from this. The bottom line is we've got big shoes to fill. There has been some really impressive work done. And I wanted to start by acknowledging the work done by Max. That has been, the PI was Roger Dietels. And the multi-center AIDS cohort here has done things like shown us over time key indicators such as the progression of HIV infection prior to the introduction of potent antiretroviral therapy. So only through doing cohorts are you able to see this was sort of the natural history of HIV, and only when you have cohort and carefully collected cohort data can you collect this kind of really important scientific evidence. Yeah, as they moved on and into the era of HAART, Max has continued to make really important scientific contributions, such as this one here, that show sort of the role of selected genotypes too as they respond to HAART over time. And the contributions from Max, when you look at the number of publications, are really impressive, and they go on in the order of hundreds of publications. The ALIVE cohort was one I mentioned before, and similar, started a little bit after Max, but not too much after, and has been following drug users over many, many years. And this was a publication back from 1995 where, again, they were able to show, and this was really important data, that there was really no difference in HIV progression for injecting drug users versus other risk groups. And, you know, there were a lot of assumptions made about drug users and whether or not you should bother trying to treat them, and this really showed that they have a similar pattern as to other populations. And then they were able to show what happens when you do introduce therapy and how that changes the mortality, both AIDS-related and non-AIDS-related, and chronic disease. You know, one of the incredible contributions of Max and ALIVE and the more clinic cohorts have been showing the interaction between HIV and chronic diseases as well as cancers. So only when you follow people over long periods of time are you able to document these kinds of effects. So besides ALIVE, they have also remained very relevant. ALIVE, as I said, was just refunded with us as a new cohort, and they continue to enroll new members into it. But here you can see that they've continued to show that uptake of HAART is not optimal for their particular population that's studied. And so they're able to sort of point to the places where work still needs to be done. And this about interruptions in therapy is very important in terms of understanding how people's HIV is progressing and also potentials for transmission. And so these sort of patterns, again, that are shown over long periods of time have taught us a lot about management of HIV among drug users. They've, as I mentioned, remained highly relevant. You know, one of the big topics in HIV prevention and treatment right now in the United States is this issue of stages of engagement in care, what they call the treatment cascade. And so the ALIVE cohort was able to sort of show where IV drug users fall along this trajectory, and you can see that the numbers really do drop off pretty significantly for those those who have sustained viral suppression over time in this cohort. Similarly, up in Vancouver, the group there has followed a cohort for a long period of time, and they have helped sort of document changes in substance use across the population over time so that you can see the patterns of which drugs are sort of the drugs of choice at that particular time in their population. They've done a lot on HIV community viral load, and again are sort of known for making some of the most significant contributions to our understanding of HIV at the sort of population level and community level in drug users. So this group up in Vancouver, we're really pleased to be working with them in the future as part of the consortium. So on that, I just wanted to say stay tuned. This is one of our new materials that's in draft materials, I should say, since we probably will change the actual images, although it'll be a similar image. And keep us in mind if you are researchers or students interested in working in the future, if you're out in the community and are interested in the science of HIV. Also, please stay tuned. We'd love to hear from people. We do have a website. I should have ended with that on the bottom. But if you look at, if you Google the M study, you'll see our website, which has information for the population in general and information about numbers of people that we enrolled. We have enrolled, we're getting closer to 100, but are still under 100 men. But we're starting our second visit, so we are off and running. So I think I'll end with that and give people a chance to ask questions or have observations from anything about our science to our materials. And you know, our materials are targeted towards our community, so they might not appeal to everyone who's online right now, but we are always open to feedback. So thank you very much.
Thank you so much, Dr. Gorbach. We have about 20 minutes for questions. If you have a question, simply type it in the field at the bottom of the chat window in the left-hand corner of your screen. So our first question is from Stephen, who says, can you say more about the difficulties of retaining participants in a study like this and strategies you've used, for example, incentives for improving retention?
Okay, it would be my pleasure. That is a tough question. I don't have a single answer. I can talk about, yes, the strategies we've used. So we have a couple things in line and planned for retention. You know, we do have dedicated staff that are going to do this. We will use technology like text messages and emails to remind people of upcoming visits. But yeah, particularly drug users are very difficult to follow over time because they go in and out of jail and sometimes even go to prison, and so that becomes a challenge. They often have unstable housing, so they change addresses. They often change phone numbers. They change emails. So you have to take a multi-pronged approach. That means that when we enroll them, we collect all this kind of information for them, and we're also using social networking websites a lot. So while people may not have the same telephone, they may keep the same Facebook account. Or their Twitter account or something. And so we're trying to find at least one sort of stable form of communication for all our participants and recognize that that needs to be tailored to each participant. So we try to work with them to find the best way to keep in touch with them. Yes, we have incentives. So we pay people for each visit that they come to, and if they need to do an interim visit for that as well. The incentives help, but they're not enough. You can't pay people with disorganized lives enough to keep them organized and remember to come in on time. So what that means is that it takes a huge amount of resources to support the staff and the sites to anticipate that many, many visits will be missed. Many visits will be scheduled and never will you see the person appear. We have learned that over time. We have done things like even book cabs and have them show up at people's houses. But you know, when you're talking about folks that are heavy substance users, that isn't enough often. We are trying to do some things. We have plans to do some work with the actual jail here to help us with retention, to help us find people who do circulate in and out of jail. So that's why we had Mark Malik, when originally we wrote this, was the epidemiologist over at the jail, and I think he was clinical medical director. He no longer is in that role, but he has a lot of experience working with the jail, so he provides us some guidance on this. And as we move forward in retention, we are just, I think we've enrolled 2 or 3 people only, or seen 2 or 3 only second visits, 6-month visits so far. But as we ramp up in retention, we will be working more with the jail on this. Then our staff know that sometimes they need to get out on the pavement and pound the streets to look for people. You know, there are known places in the community where some people sort of hang out. So some of these techniques can be used to sort of track people if we really have trouble with retention. I should say the cohorts that have done this in the past, like Max and Alive, have had very good retention rates because they have established relationships with their cohort members. So that's a lesson we have taken from those cohorts that are long-term, that you need to develop a relationship of trust. and develop a relationship that enables people to find you when they want to find you. So we hope to also learn from the experiences of the other cohorts in our consortium. You know, the folks at Alive that have been working with drug users for many, many years are really experienced in this and have been very helpful to tell us sort of how to do this. So, you know, that's sort of our general specific strategy here, and also some of our general strategies.
All right, fantastic. Our next question comes from Eric, and he says, it may be too early to tell, but is there any good news on the horizon about illicit drug use declining in these communities? This week, a new poll from Wall Street Journal, NBC News shows record support for same-sex marriage at 59%. And specifically, support has grown markedly among Hispanics, women, and people who lack college degrees, among other demographic groups. One assumes this extends to approval of gay people in general. You'd expect this to eventually impact the groups that you're looking at, correct?
We would hope, and it has been really exciting to see the changes in attitudes in general for people that are gay identified in the United States and in the community. Right, and as part of that, you know, we, when in our own research team, some of the old measures that have been used to measure sort of the impact of feeling of being gay in the United States, such as homonegativity, we've realized need to be reframed as sort of homopositivity, that people feel good and are feeling more and more good about themselves being gay, and recognize that people in the community, in the communities they live in also are more positive towards them. So that's something we are hoping to track the effects on over time, and recognizing that you know, the landscape is changing. Whether or not and how this affects drug use is something that we will be really excited to be able to contribute to measuring. So we hope that it will result in different patterns of drug use in the community if use of drugs has been associated with sort of these old homonegativity measures, and that as people are more positive, they'll turn less to substances. And since we're starting sort of right on the cusp of this important historical moment, we hope to show that. Let's be positive and optimistic and hope that there'll be less drug use. But, you know, as I showed you, drug use is something that is a sort of basic human drive. People use drugs. They have used drugs throughout history and throughout time. The question is what drugs they use. And I think what we will be documenting is maybe a transition to different drugs and different patterns of drug use, but there will always be drug use. I should mention too, we are measuring heavy alcohol use as well, and this remains you know, constant problem in our society and in human populations in general. Tobacco use as well. We are measuring e-cigarettes as new tobaccos and other sorts of new technologies of substances as they're introduced. So that's part of our job, and that's what part of it will be exciting, is to look at these trends and how they impact HIV and also sort of the general lives of folks in the communities that we follow over time.
Great. And our next question comes from Heather. She asks, what are your plans for participants who enter drug treatment during the course of this study?
Okay, Heather, great question, and we really hope that our participants will enter drug treatment and they will continue to be followed. I didn't include a slide on it here, but in the last cohort I did of newly HIV infected men who have sex with men, in which we followed them only for a year, we were able to track the trajectories of drug use, and we noted that you know, there's sort of three obviously general patterns. People either remain stable with their pattern of substance use, they use substances less, either through starting their treatment for their substances, or just deciding to stop, or they initiate substance use. So we expect to see people in all of those different patterns. And one of the things that we will be able to contribute is understanding the factors that are associated with people who move in and out of these different patterns. Now, people who get treatment for substances often come in and out of treatment. Substance use treatment isn't always a straight clear trajectory path. That means once you get treatment, you're done. We expect to be able to follow people in and out of treatment over time. Once they start treatment, that does not exclude them from being in the cohort. They will be followed. So we will, again, work with the treatment centers and work on trying to retain people if they're in inpatient treatment, but it's very important to us to document patterns of treatment and how that affects sort of their general health. You know, we would hope to see that people who were non-adherent maybe to HIV medication before starting treatment maybe have better adherence after starting the treatment, but those sorts of patterns we will try to document and look for.
Thank you so much. Do you have anything else to add, Dr. Gorbach?
I don't. So I hope, I mean, I could talk forever about this, but I hope that sort of gave a broad strokes overview of what the platform is and how we try to do it, and also provided you with some evidence for why it's important for us to be doing this here at UCLA. So I look forward to hearing from many of you in the future.
Wonderful. Thank you so much for joining us, and thank you, Dr. Gorbach, for your time. Please join us next month when we continue the conversation with Professor Hilary Godwin. She will talk about building resiliency towards climate change in Los Angeles, a collaborative approach. If you missed any of our previous webinars, go to the School of Public Health's website, which is ph.ucla.edu. They're all archived there. Please allow a few days for this one to post. Thank you so much.